So kicking off today's session is going to be Dr. Shan Xu. Shan, please feel free to come online. So Shan is currently a senior scientist and the associate director of the focused ion beam and scanning electron microscopy technology at Janelia Research Campus. Shan comes to us from actually the semiconductor field, and today he'll be telling us about how using FIB-SEM technology is able to reveal a brand new world of cells at isotropic, EM resolution down to about 2 nanometers or so. So, Shan, the floor is yours.
Okay. Can you see my screen here?
Yeah, Shan.
Okay, great. Thank you, Goku, for the introduction, and really thank the organizer for inviting me to share my work here. Wish I could be back to Berkeley. I had a lot of great memory there. Anyway, today I would like to talk about a new form of a microscopy that's based on electrons, not photons, that what you have heard in the past 2 days. It's called FIB-SEM, or focused ion beam scanning electron microscopy. Of course, we are not looking at live samples anymore with EM, but I hope these frozen in time snapshots, you can see on the bottom here, provide a new perspective on how we visualize the cellular world at the nanometer resolution and in the context of whole cells and tissues. So my talk is going to be divided into 2 parts. I will first introduce the motivation and development of the this enhanced version of FIB-SEM, and then I'll show you a few examples of what value this newly developed technology can add to the biologist. So, for example, if you want to study organelle structures and interactions, depends on how many labeling colors you have using a fluorescent light microscopy. You can only see maybe a few of them organelles at a time. Now, if you switch to electron microscopy, for example, like what I'm showing here, it's a electron micrograph of a macrophage. You immediately see quite a few things, right? Of course, the plasma membrane, and here's a big nucleus, a very beautiful chromatin that's labeled here, and a variety of organelles as far as they can be labeled through this EM staining. And luckily, these lipid can be stained nicely with osmium, this heavy metal commonly used in EM microscopy. Maybe not quite easy to see it here at this zoom out level, so you could segment out these organelles and color them like this. So the bottom line here is for this EM technology, you immediately get, number one, multiple organelles showing up at the same time, and number two, you have nanometer resolution. But the question is really, how can you do this, showing all these organelles in the whole cells, and even better in its native tissue environment in 3D? So one of the answers is to use FIB-SEM. So what is FIB-SEM? Well, FIB-SEM is also called the cross beam because it has 2 beams, a scanning electron beam coming from the top and the focused ion beam coming from the side. To acquire a 3D volume using FIB-SEM is rather straightforward. So you first take an image of the top surface using the scanning beam, and then the focused ion beam coming from the side mill off just a few nanometers of that top material. And you just basically repeat that imaging and milling cycle over and over for like tens of thousands of times until the entire volume is imaged and milled away. So it is a destructive method. The main reason why we choose FIB-SEM is that it provides these beautiful isotropic voxels, which is important, as you can see here, for example, like the neuronal tracing. And later on, if you want to do auto segmentation, it has a lot of great benefits. But resolution is not the whole thing. We still need to consider volume, size. What I'm showing here is a working space of that can be easily compare different volume electron microscopy techniques. On the y-axis is the best isotropic resolution, in nanometers, and on the x-axis is the size or volume. They're both in log scales. Now, as you can see here, this is a big, a wide open space that's not covered by any commercially available technology. But I think that's probably what most biologists want, because it provides, say, like less than 10 nanometer resolution, and the volume here is about 100 micron cube that consists, you can hold multiple cells in that volume. So commercially available FIB-SEM, even though it has this great isotropic resolution, but the size is kind of limited. Only about, you can use that image at 8 nanometer, for example, about the size of each cell. So 10 years ago, Jerry Rubin, the founding director of Genelia, has this grand vision to image the entire fly brain, right? So, but the fly brain volume is 40 million cubic microns. That's like 4 orders of magnitude bigger. This is a big gap we need to fill. So why these commercially available systems have these limitations in terms of volume? The main reason is that there's a long list of interrupts, right? I'm listing it here. And it's really challenging to restart after these interrupt without a data loss. Because we're talking about nanometer, not only in the imaging, but also for the fit milling, which is disruptive, as you already realized that. And on top of that, you got a slower imaging rate from SCM, and then a system normally could drift within a few hours. So what you end up is just a kind of small volumes with a few days of operation. So how do we solve this problem? Well, long story short, I redesigned the entire control system and developed what I call a smart architect that's immune to interrupts. So my philosophy was to create a smart system with 100% effective reliability that's not afraid of these interrupts. And most importantly, is capable to resume the imaging from these interrupts without defects in the final imaging stats. So besides the architect change, there's a long list of packages and features which I won't have time to go into details, and they're all published. If you're interested, we can talk offline. Just highlight a few of them. So first, we reposition the FIB column to be 90 degrees, so that provides better and stronger signals so we can image faster. And also implement a closed loop fit milling control. And this is really crucial. That provides a precise milling during normal operation, as well as a smooth re-engaging after any of these interrupts. I also have this inline focus, autofocus scheme that has no overhead, so keep the image focus, et cetera, et cetera. So at the end, really, now after 10 years of hard work, we have a system that can image fast and can operate continuously for months to years. With this capability, we are ready to tackle the fly connectome. So here's what we call the hemibrain that can consists of 16 million cubic microns of a volume. Our colleague Ken Hayward first partitioned this brain volume into 13 slabs, and then we can image them in parallel in 2 machines over 2 years on and off. Now what you're seeing here is the seam that we digitally restitch back these 2 tabs that were imaged on 2 machines individually. And the goal here is to trace the neuron across both these seams and across this entire volume, every single neuron. That's what we call dense reconstruction, not just a sparse picking out a few neurons. So as a matter of fact, you can see a lot of features here, right? Mitochondria, for example, besides the neuronal tracing. In fact, there are more than 6 million mitos here. Now, over those 2 years operation in these 2 machines, we have literally more than 100 interrupts here, but you don't see a gap in the final volume. And that's what I meant by 100% effective reliability. This is really crucial for this dense reconstruction of the fly brain. So with the help of Google's and 50 proofreaders at Genelia, now we have obtained this largest connectome today with more than 25,000 neurons and 4,000 cell types. just put the volume into perspective, right? So this is the volume of the fibrin, 16 million cubic microns. What I'm showing here on the left upper corner here, the little red cube, is what a commercial system was able to do 10 years ago. And unfortunately, still the case today. So the bottom line here is really with better image quality, better segmentation, improved reading tools, we achieve a couple orders of magnitude improvement in the connectome pipeline throughput. Not just the imaging, it's the whole pipeline. That's what we really care about, compared to where we started 10 years ago. So everything here, both the raw data and the segmented version, is available open to public. You can go to newprint.genelia.org, the website, to browse it at your during the free time that you have. So now we have a new platform called Enhanced FIB-SEM. Instead of having a small volume like this, now we have a much expanded working space and easily to capture the entire fibrin. And you can imagine many more applications could utilize this newly available capability. So in the remaining time that I have left, I will show you 3 more examples that demonstrate the unique capability of enhanced FIB-SEM. The first example is mouse pancreatic islet beta cells. This is a collaboration with Andreas Miller and Mikel Salamana. So we know that the main function of these beta cells are to synthesize and store insulin in secretory granule, these little black circles here, and to release them under a high glucose stimulation to maintain the blood glucose homeostasis. So what is the structural basis to fulfill such functions? And particularly the role, what does this the microtubule play? And it's still under debate. One could imagine microtubule can act like these transport tracks to pull these secretory granules to the plasma membrane for exocytosis. But it's also possible that a very tight connected microtubule network hinders the transport of secretory granule at low glucose condition. But somehow this network loosens up at high glucose so that allow this secretory granule to reach the plasma membrane for release. So to answer this question, we first FIB-SEM image multiple beta cells that are prepared by Andreas. And you can see here, is that movie playing? Okay, great. Then Andreas and his collaborators segment out the raw EM volume, segment out the microtubule, mito, Golgi, ER, ranging from fully automatic segment to semi-manual and the fully manual. But at the end, you get this beautiful segmented version of the cell. As you can see, secretory granule is the majority of them. It's colored in orange. There are actually 5,000 to 10,000 of them in one of these cells. Now you're seeing the green Golgi, microtubule, the main characters of this study. So after the cell is segmented, we image total 7 cells, as I said. 4 of them are high glucose condition, and 3 of them are low glucose condition, so that they can do the comparison. So with the after segmentation, Andreas performed a series of this comprehensive and quantitative spatial analysis of all these cells. Here is just one of the analyses as an example I'm showing here. It's the microtubule, a length and curvature comparison between the 2 conditions, low glucose and high glucose. As you can see, when the glucose concentration goes up, the microtubule length substantially decrease, but the curvature still maintain the same. So long story short, the overall finding we can summarize in a cartoon form like this. In low glucose conditions, we have very long microtubules, kind of tied to the secretory granules near the membrane. And under high glucose stimulation, these microtubules kind of fragment out into small pieces. And at the same time, we saw this increased amount of secretory granule near the Golgi. So perhaps the role of a microtubule is simply to secure these vesicles near the membrane, get ready for release under some stimulation. The second example that highlighting the application of the FIB-SEM beyond connectome is about structure and functions of hepatocytes. So our collaborators, Gunesh Palagu and Anna Aruda from the Hatamishli lab at Harvard hypothesized that the functional diversification of these hepatic ER is due to its structural regulation. So in order to answer this question in its native tissue environment and in the context of a metabolism, they want to obtain detailed 3D information of the ER structure and organization in the tissue, liver tissue, under physiological and pathological states. So we start by imaging multiple tissue samples of different conditions that Anna and Gunesh prepare. We have lean versus obese, fasting versus fat, and they're all large enough, as you can see, to contain multiple whole cells. And you can already see the morphological difference between, say, lean and obese, which becomes more obvious after segmentation. For example, you can see a lot more lipid droplets here, showing in yellow for the obese animal, which is not surprising at all. And we can zoom in to look at really the difference, say specifically on the ER structure between the lean liver versus the obese. As you can see in the lean liver, you get this beautiful ER stack sheets, just very nice looking, just like the maze. But on the other hand, with an obese animal, most of those sheets turn into tubules, as you can see, like a nice little tunnel going through. And Gunesh and Nana then quantify this striking remodeling ER structure. And to look at, for example, you can see the ER sheet volume decreased down to about 1/3 in the obese compared to the lean animal. And also you can look at the ER sheet to tubule ratio also dropped by more than 50%. But the story didn't end here. Ganesh and Anna wanted to find out if the metabolism function can be recovered after the structure is restored. So they used this CLAMP63 protein, which is a membrane-shaping protein. And as you can see here, after applying this protein, you can kind of restore the ER sheet already. And of course, it can be confirmed just from the raw EM data, and also can be confirmed through quantitative analysis. And they further substantiate these findings, the structural finding, with an array of biochemical and fluorescence imaging approaches. At the end, what they have found or show is actually the metabolism function also recovered. As you can see here, the glucose concentration dropped both dynamically and statically with the help of a CLAMP63 protein. Once you restore the structure, the function also returns, which is really amazing. So far I've shown you a few examples about tissues with cells containing, like the brain, more than actually tens of thousands of cells. You can also go into the other side of that expanded space, zoom into a smaller volume at higher resolution. And that's exactly what we are doing here. What I'm showing here is an entire HeLa cell, a culture cell, image at higher resolution, 4 nanometer. By the way, the previous study was mostly done at 8 nanometer voxel size. So I have to admit, it's substantially more challenging at 4 nanometer in terms of system stability and reliability. However, this rich information, you can see all these organelles offered by this high resolution data sets is invaluable. And you will hear from Aubrey in the next talk on how she and her team actually lift these black and white image data sets to the next level. So instead of staring at one cell for hours here, it probably would be more interesting to look at 2 of them together and see how they interact. And that will be my last example. It's a T-cell attacking cancer cell. This is a collaboration with Alex Reeder and Ira Melman from Genentech. So as you can see, the T-cell color in green and the cancer cell color in cyan color here, the interface between them is the immunological synapse, which is the close contact between these 2 cells. And this is really the battleground where the critical and interesting actions occur. And it's the perfect application for FIB-SEM because the isotropic 3D imaging can easily capture this entire curvy and wavy immunosynapse surface and its surroundings at 4 nanometer voxel size. More importantly, the enhanced FIB-SEM can capture a volume that's large enough to that include both, not just one, both of them, in both cells. So I guess just to summarize, over the past 10 years, we have transformed a conventional FIB-SEM from a lab tool lacking of long-term stability to a robust imaging platform we call enhanced FIB-SEM, capable of years of continuous imaging and without defects in the final image stack. So although the enhanced FIB-SEM was initially motivated by the fly connecting challenge, the end result is a transformative imaging platform that provides a sweet combination of resolution and volume that enable many discoveries, from large brain tissues contains tens of thousands of cells to a single cell at high resolution atlas application. So we're really excited about this unique capability and think this is just the beginning of a new field and a holistic way of looking into the cellular world. With that, I would like to thank everyone that contributed to the work that I'm presenting you today, particularly Genelia, the FIB-SEM technology group, and the HAS lab for the development of the technology, the FLY-EM team led by Steve Plaza, the COSAM team led by Aubrey, which you will hear a lot more in the next talk. Of course, I won't have these beautiful biological story to share with you with fantastic external collaborators. The mouse pancreatic beta cell example from a collaboration from with Andreas and Mikkel, and as well as the liver story from Ganesh, Anna, and Gokhan. And finally, the T cell attacking cancer cell story with Alex and Ira. And thank you for your attention. I'm ready to take questions.
Thank you so much, Sean, for that amazing and beautiful talk. For the audience, please feel free to type the questions into the event chat, and I will read them out. But Sean, I have actually a few questions that I wanted to go through with you. So over the past few years, there's really been growing interest in bringing FIB-SEM technology to basically a lot of research institutes. And most of them base some of these decisions based on some of the data that Genelia puts out along with you and your collaborators. But one of the things that most people don't realize is the amount of effort and work that went into taking a commercial instrument and modifying it such that you now have optimized milling and imaging angles, and you have optimized imaging time such that after the 2 or the 3 day runs, you can basically restart for larger samples. Now, most commercial instruments that I'm aware of may not, readily address some of these challenges. So what is your advice to, say, departments that are considering getting some of these instruments now? How would we kind of go about getting access to the same level of robustness and stability that you have built at Genelia?
That's a good question. Actually, the good news is this whole technology package, or you can think of it's open at Genelia to free license to any non-profit organization, including universities or research institutes. So we already have, because we have a patent, we have an entire package, build-up material, software go with it as well. And as a matter of fact, LMB, MRC LMB, they already have 2 of these tools, and they license our technology, and they're in the process of adding this package onto the commercially available system, because we feel like this is a very, I call it production-worthy solution, because most of the time people just do it once or twice. Here at Genelia, actually, we have like 12 of these systems. I built it up over the years. So they all kind of, you can consider as like a production, like these semiconductor fab running 24 hours day and night. So I would recommend either contact me or probably straightforward, or you can also go to the Genelia Open Science website to look at the technology licensing options.
Perfect. I have a few questions from the audience members here. I think William, Sean just probably answered your question about these enhancements making their way to the commercial systems. Hannah's asking about how do you segment different organelles, but I'm assuming we're going to hear a lot about that from Aubrey. But Sean, if you have more comments, please feel free to add to that.
There are actually a lot of options in terms of segmentation. For example, our Fly Connectome projects, and that we have a good collaboration with Google. And so there are individual labs, they do segmentation as a development. And also there are like here, the CoSAM team. And also there are commercially available packages or options. You can, I probably shouldn't name it as an advertisement, but there are options. You can pay these pay service, you can get your data segmented out. And of course you can go with manual. For example, like a good example for this is the pancreatic islet data set. Andreas actually did 3 different kind of segmentation. One is fully automated with collaborator. And then the second one is a kind of semi-manual. And the last one, the microtubule, which is very hard. So he did it manually. So you can always go through brute force to do it manually.
And then the last 2 questions from Adila. She's asking, are lipid droplets causing the ER fragmentation in one of the studies that you've shown with Gokhan's lab?
So could you repeat that question again?
Are lipid droplets causing the ER fragmentation?
That one, you know what? I'm not a biologist. I'm not sure whether that is the case because the lipid droplet, my guess is it will play certain roles because the interesting challenge here is oftentimes if we just focus on one specific organelle, and it might miss the whole picture because now we have the opportunity to image the whole cell. You can segment out every single organelle, so you can see, okay, for this specific phenomenon, is it just the cause is from a single organelle or maybe 2 or multiple of them, the combined effect at the end. So I would probably defer this to our collaborators.
And the last question is from CMAC, who's asking, why did you need to or decide to use 2 FIB-SEMs in your original FlyBrain work at Genelia?
Okay, at the time we only have 2 FIB-SEMs. That's a straight answer, but literally when you have a whole FlyBrain, the volume is so big. If you want to run it in a single machine, it can take much longer. So the easy way, at least the approach we have been taken, is partition a big volume down to slightly smaller volume. In this case, we have like 13 tabs so that you can run things in parallel. As a matter of fact, right now we are running an entire Fly CNS with 8 machines in parallel.
Thank you so much, Sean. I would like to be the person that's giving you the applause on behalf of all the audience members that has truly appreciated your talk. Thank you again for joining us.